First of all, I want to thank everyone for joining us today. And of course, I'd like to welcome you to today's presentation of the Congressional Biomedical Research Caucus. The caucus provides a forum where members and staff can interact directly with preeminent researchers responsible for important scientific discoveries. We encourage strongly for your boss to join the caucus if he or she has not. You can contact Congressman Curtis, who has joined us today, who is one of our 4 co-chairs, along with Congresswoman Jackie Spear, Congressman Steve Stivers, and Congressman Stephen Cohen. And it is because of their hard work and commitment that we've been able to host these briefings for 32 years. That being said, Mr. Curtis, I would love it if you had a few words that you could share with the group, and then I will introduce Dr. Yamamoto.
I would love to, but I'm going to take you up on a few words and just simply say it's a delight to be with you. Doctor, thank you for joining us, and to those who are a part of this, thank you as well. And you're absolutely right. Would love to have anybody join this caucus, and it's certainly an important one on the Hill. And with that, I will turn it back.
Great. Thank you very much. So today it is my pleasure to welcome Dr. Keith Yamamoto. Dr. Yamamoto is Vice Chancellor for Science Policy and Strategy, Director of Precision Medicine, and Professor of Cellular and Molecular Pharmacology at University of California, San Francisco. After earning his PhD from Princeton University, Dr. Yamamoto joined the UCSF faculty in 1976. His research has focused on signaling and transcriptional regulation by nuclear receptors. He uses mechanistic and systems approaches to pursue these problems in pure molecules, cells, and whole organisms. He has led or served on numerous national committees focused on public and scientific policy, public understanding, and support of biological research and science education. He chairs the Coalition for Life Sciences, co-chairs the National Academies of Science and Engineering and Medicine Roundtable on aligning incentives for open science, and sits on the Board of Directors and Executive Committee of Research America. He's on the Board of Directors of the Public Library of Science, the Governing Board of the California Institute for Regenerative Medicine, the Board of Counselors for the Radiation Effects Research Foundation, and the Advisory Board for the Lawrence Berkeley National Laboratory. He has chaired or served on many committees that oversee training in the biomedical research workforce, research funding, and the process of peer review and the policies that govern it at the National Institutes of Health. He was elected to the National Academy of Sciences, the National Academy of Medicine, the American Academy of Arts and Sciences, and the American Academy of Microbiology, and is a fellow at the American Association for the Advancement of Science. I know we are all looking forward to hearing you speak today, Dr. Yamamoto. So without further ado, I turn it over to you.
Thank you, Lynn. And thank you, Congressman Curtis, for your leadership of this caucus. And I agree with you that it's an important one. And we hope to have as many members joining as a result of this caucus and others that the Coalition for Life Sciences helps to put together, and look forward to a robust discussion. A very alliterative title, Priorities, Preparedness, Precision Peers, and Payments to Enhance Health. That's really what the NIH, the National Institutes of Health, is all about. And we're going to discuss exactly that. It's funny, it's not advancing. How's that? And the NIH has been called the crown jewel of federal spending. It's an agency that turns discoveries into health. It seeks fundamental knowledge about living systems and applies that knowledge to enhance health and lengthen life and reduce illness. It has a big budget, invests billions of dollars, $41.68 billion in fiscal year 20, to support biomedical research. That's a lot of money, but as Mary Lasker said, if you think research is expensive, try disease. And that 41 billion, while it is a lot of money, it's useful to just keep in mind that it's about 1% of the healthcare spend in this country. About 80% of that 41 billion goes to your districts, your states, to about 2,500 universities and medical schools, research institutes, and a whole pile of companies that really enable that work to go forward. In 2018, the last where I have good data, employing some 425,000 individuals in your districts and states and contributing $80 billion to your local economies. So that 41 billion is essentially doubled as it goes out into your districts and states. So how does this really work? How does the NIH work to turn discoveries into health? And at one framework, at the kind of operational framework in which all of you operate, the answer is that there's a big bureaucracy. The NIH is a loose federation of 27 separate institutes and centers. One of the agencies is the Public Health Service, which, as you know, is part of the U.S. Department of Health and Human Services. These institutes, as you scan through them, you'll see things that you recognize: the National Cancer Institute, the National Heart and Lung and Blood Institute, the National Institute on Aging or Drug Abuse, and so forth. The topical institutes with specific missions. And also we're going to discuss the little guy down here in green, the Center for Scientific Review, which oversees the peer review mechanism that actually allocates that $41 billion out into investigators, scientists, and trainees in your districts and states. So each institute receives its own budget from Congress, from you, but the question is then, once that money comes in, how do we actually prioritize the spending? And that's a difficult hill to climb, because, in fact, we really understand still very little about how biology actually works. In big framework, the answer for how the budget is allocated is here, and that is that there's a pyramid in which the work of the NIH is really based on a very solid foundation of basic research, fundamental discovery about biological processes. As Vannevar Bush said in 1945 in this ageless report that he sent to President Roosevelt or President Truman, basic scientific research provides scientific capital. And the priority then is to discover fundamental mechanisms of biological processes and to use untargeted research to understand, treat, and cure disease. So that foundation of basic research then supports translational research that bridges the fundamental findings to clinical research and clinical health care. And then there's at the top of the pyramid then is a small amount, a relatively small amount of projects that are supported directly in clinical research itself. It's important to keep in mind that the federal government must support this great important foundation of basic research, because industry can't. Industry really needs to keep its head down, keep focused on specific projects and that will turn into products that it can sell, use to make a profit, and of course use to help people's health. So the industry really operates in this upper part of the pyramid, and the fundamental knowledge that drives all of that is really dependent on basic research supported by the federal government. So why does it work this way? And why should there be this kind of big mass of fundamental discovery that's just asking, which scientists are simply asking curiosity-driven questions about how biological processes work? And the answer is that why it's so complicated is that biology itself is complicated. And it's complicated for a good reason. So here's some information that all of you learned in high school that I'll just kind of go back and remind you about, because it forms such a critical part of how the NIH works and the approaches that it's taking. So here's just a set of simple pathways, biological pathways. This one is unidirectional, going from fertilized egg to an embryo. The other two are bidirectional, fat breakdown versus fat buildup, non-dividing cell to a dividing cell, and vice versa. These are processes that are encoded in our DNA. But as we'll see, the processes themselves, as they play out in you, are not hardwired. They're not just simple switches or even long multi-step linear pathways. Because if they were hardwired, we wouldn't be able to respond, to sense and respond to changes, differences in our physiology or in our environment or in our life experiences, in our behaviors, responding to social determinants of health and so forth. And so that's not going to work. So instead, what we have is this, that there are networks of interacting proteins that alter the expression of genes. So if you look at, as you look at this plate of spaghetti here, what you can see is down at the bottom are the genes that are actually driving biological processes, or in some cases, unfortunately, disease processes. Those genes are like light bulbs on a string that can either be lit or unlit, or actually lit brightly or lit dimly or turned off completely. All right, and how strongly, and whether the genes are turned on or turned off, and how strongly they're turned on and turned off, is determined by all of this plate of spaghetti above the genes themselves. That's and that whole diagram is that. At the top are the key things, that there are sensors, antennae, that are receiving signals, physiological signals within your body, environmental signals coming in from the outside, that trigger these various complicated interactive signaling pathways that go through the cell, and eventually trigger the activity of proteins that then serve to activate, to turn on genes, turn on strongly or weakly, or turn them off. So those genes that are hardwired in all of your cells, in each of your cells, right, are controlled by this big network of interacting factors that affect, and it's the reason that each individual is different, and that it's the way that these signal receptors are triggered, and the way that they then trigger these various pathways differs from individual to individual. So the result then is that biology is complicated, and therefore so is disease, because disease is just mutations or alterations that affect the way that these various sensors are actually working. So they in turn then affect the risk or course of disease, and it means that genes for any of these factors on the diagram could end up being disease genes, in the sense that they will affect the risk or progression or outcome of disease if you are unfortunate enough to be stricken with the disease. So that's why biology and disease is complicated. A corollary to this great complexity, right, is that a lot of the fundamental knowledge is uncovered often in simple experimental organisms that scientists use in our laboratories. So simple organisms like those cartooned here or photographed here contain fewer layers of these sensors and regulators. So the plate of spaghetti is not as complicated to figure out. Simple organisms allow simpler, more definitive experiments that we can do in the laboratory that through biological complexities or simple ethics, we wouldn't be able to carry out in humans. And that complex organisms, that is you, evolved from simple organisms. So what we learn in those organisms is instructive for what we can now understand about you. That process, that progression, starting with simple systems where we can understand things more easily, really works. And you can look here at this compendium of the sources of big breakthroughs in biology. I won't go through them, but it's actually more interesting and amusing, in fact, to kind of scan the experimental organisms that have given rise to these key pieces of information about biological processes and disease that range from bacteria and their viruses, the viruses that affect bacteria, not you, yeast, clawed toads, chicken viruses, fruit flies, sea urchins, my favorite, pond scum, sea snails, and so forth, where really critical secrets about the way that biological processes work have been uncovered. But wait, you might say, you know from what we've all experienced in the last year, that COVID, that research on COVID, and the therapeutics and diagnostics that have been developed in record speed by scientists working directly on the disease, directly on the virus. So what's this nonsense about having to go through this complicated progression of experimental organisms? Doesn't the rapid progress on COVID mean that we don't actually need all this basic research? Or maybe we've done enough that we understand enough that we don't need any more, right? So let me just disabuse you of that point of view, which you may hear as you go out and talk to your constituents in your districts and states. So what we know is wonderful, and that is that when COVID struck, researchers, scientists all over the United States and all over the world, but certainly NIH-supported researchers, many of them pivoted their work, and they switched from the projects they were working on to being able to use their expertise and their technologies and methodologies and concepts to be able to work on the virus itself, SARS-CoV-2, and on the disease that the virus causes, COVID-19. And in just a few months, those scientists were able to, and companies that they worked with, were able to create incredible set of diagnostics and tracing methods, therapeutics, remdesivir shown here, but that's just one of now a number, and these miraculous mRNA vaccines that are so remarkably efficacious. And so knowing all of that, people in your constituents may be saying, so enough with the basic research already. We just work directly on the disease, directly on the virus, and we nail it. And I think that we're now well on our way to being able to take care of this pandemic. Still a lot to be done, but we're definitely well on our way. But in fact, those months of effort were actually founded upon a huge, long history. So the targeted success with COVID really depended on decades of untargeted basic research. I'm not going to go through all of these, but for each of these, then I've actually just listed a few of the basic discoveries that enabled these applied discoveries to be able to go forward. And you don't even have to really worry about or look at them. We can discuss any of them that you wish, but maybe it's just instructive just to look at the dates in which these fundamental pieces of information were uncovered and see what I mean by decades of basic research that enabled the applied work to be able to go forward. So it really means then for future preparedness, we really need to maintain this background, this foundation of science and technology. So in a way, we can think about the COVID experience as a wake-up call that says that we need not only to maintain, but to increase our support for science and technology. We know that, in fact, the response in the United States to the pandemic has been halting and complex and difficult, and could have been better enabled if we had been better prepared. And it's really interesting that Research America carried out a poll just before, toward the end of 2020, just before the big surge, the big post-Thanksgiving surge that the country went through with such pain and loss. But even at that time, before that surge, a public opinion poll taken by Research America uncovered the fact that the strong majority of the public, 79% of the public, said that this pandemic is a disruptive event that requires urgent refocusing of America's commitment to science. So voters in your district say that it's important to step up the game for scientific research. Americans support the importance of increasing the percent of GDP that goes into research. That there are governments around the world that are putting 3 to 5% of their gross domestic product into research and development. Some have already gotten there, others are well on their way. China is increasing its R&D budgets by double digits every year. How important is it for the United States to be able to do that? Again, about 80% of Americans say that it's either very important or somewhat important to do that. For comparison against that 3 to 5%, look at what the current level of US research and development spending is, 0.7% of GDP. We're a long way from that 3 to 5% goal. I've been associated with a group that generated a science and technology action plan that you will be hearing about, because we are just moving this into discussions with Congress right now. In fact, I had a couple of Zoom calls in the past couple of days to discuss this plan and being able to move it forward into legislation. So you'll be hearing about it, and I just wanted to make sure that you were aware of it. Here we go. In a poll taken like a couple of years ago in January 2019, again, Research America, it really appears that the public understands the link between basic research and application, in the sense that they really support federally funded basic research. Here's the question, and this is exactly how the question was worded, so I think it's instructive. Even if it brings no immediate benefits, basic science research that increases the frontiers of knowledge is necessary and should be supported by the federal government. 80% of citizens strongly or somewhat agree with that statement. So as you interact with your constituents in your districts and states, it's important to kind of keep in mind that at least nationwide, there's very strong support for research and for actually continuing this investment or extending it. And I think that's a really key thing to keep in mind. So now we can really ask, you can ask or should ask the key question. Is that, okay, that's good that the public understands this. Is this really the right way to be doing it? Does it work? Does untargeted basic research eventually bring benefits? And we can say uncategorically, the answer is yes. Here's one version of the yes answer of the contribution of NIH funding to new drug approvals. Over this 7-year period, 2010 to 2016, the FDA approved 30 new drugs per year, right? 2 to 3 new drugs per month were approved by the FDA, 210 new drugs were approved over that period. Now let's look at those, whether NIH basic research contributed to the new drugs that came forward to the FDA and were approved and now out on the market, sitting in CVS and Walgreens. NIH-supported research associated with all 210 of those new drugs. And 90% of that NIH funding was to gather basic research data. So there's the link. And it really shows then that NIH funding enables industry research and development to develop therapeutics that end up being effective in increasing the health and well-being of your constituents. So then the question is whether we can be doing better. Could we really bring together all of this basic research information, all of the stuff on fruit flies and yeast, together with all of the clinical studies and population studies on real people, your neighbors and constituents, to more efficiently and effectively move health forward? And that's what precision medicine is about. You may have heard of that term. The Precision Medicine Initiative was announced in 2015 by President Obama and has been continued through the Trump administration. So what the heck is precision medicine? Precision medicine, again, a large framework, you could think of as a Google Maps for biology and disease. But instead of stacking different geographic features on top of each other to be able to correlate, where the Starbucks is relative to the railroad, instead, we break different data types, biological data types, into layers and stack them on top of each other, and then look for correlations between the different layers to build what is called a knowledge network. And that knowledge network then is really the goal of precision medicine, being able to link different kinds of information in ways that can be informative about people and people's health, to build a data network that aggregates and then analyzes information from patient cohorts, from healthy populations, from experimental organisms that we looked at, to define disease mechanisms and disease networks. And from that, to be able to provide precise health and health advice, diagnosis, and treatment down to the level of individuals, not statistical groups of people, but individuals that really highlight that we know enough to be able to see how one person's risk or progression of disease is going to differ from others in ways that can be predicted for treatment or hopefully even cure. So the promises and challenges of NIH research are many. The outcomes of precision medicine as we move toward it will be a research ecosystem that makes full use of discoveries, those basic, fundamental, curiosity-driven bits of information, that health and health care will become tailored to individuals, not groups, and hopefully accessible to all individuals. The higher quality of life and greater work productivity that will come from that, and that this is really probably the only way that I can imagine that we will be able to reduce the slope of the healthcare curve, reduce healthcare costs, through improved prevention, earlier diagnosis. We all know that the earlier you can diagnose something, the better the chance for a positive outcome. Better control of chronic disease and avoidance of unnecessary tests and ineffective therapies. Those last two things are major drivers of healthcare costs, that doctors prescribe medicines where they don't actually know if it's going to be helpful for the patient, and in some cases, unfortunately, can be harmful. But instead, they diagnose them broadly and knowing that it'll work in some cases and it won't in others. And those, of course, are major drivers of cost, just like giving MRIs to people that don't actually need them. So the priorities then are here: to study biological processes where they're best understood, the pond scum and the fruit flies, to be able to create a knowledge network that links all of that information together so it's useful for people, and then to use that information to inform diagnosis and treatment decisions for each individual, and to empower further research and to advance clinical care and inform patients and citizens. So that's good. So then if NIH is taking on this kind of big task list, who's making the decisions? And here we begin to move into the area of peer review. Who makes those priority decisions about how the money is spent? And remarkably, it's the scientists that do it. They're the ones that are at the front line, boots on the ground with the biological problems and health problems. And how the scientists do it is this process that you've all heard of, but I'm going to give you some detailed information about, called peer review. So what the heck is peer review? How does it work? So the NIH has many different research grant mechanisms they offer to the scientific enterprise and say, here's some money, come and get it. And the best of those mechanisms are the open invitations for the scientists to dream up a problem that he or she wants to work on, really fully at their discretion. And to send in a grant application describing that problem and the approach that the scientist is going to take. And then it's the scientists then who evaluate the scientific merits of those proposals. So 80% of that $41 billion that the NIH gets is allocated in this way and goes out into your districts and states to institutions and scientists in your areas that are working on these problems. So it's the CSR, the Center for Scientific Review, that I highlighted in green in one of that earlier the discussion of the organization of the NIH, that is independent of the institutes themselves, and they manage over 80,000 applications per year that come to them. They have to oversee the review, the peer review of that process. The reviews are executed by committees called study sections. You see a picture so you can get a little idea of how many people we're talking about in an individual area of study. These are committees of experts, scientists, extramural experts that are coming from your district to Washington to sit down and oversee the evaluation of these grant applications. So they're made up, the study sections are made up of working scientists who volunteer to serve, to set aside their own work to serve in this way. There's 5 criteria that are used for evaluating every NIH grant application, looking at the impact. How much difference will it make if the proposal that the scientist has put forward actually turns into experiments that work and advance our knowledge? What is the experimental approach that the scientist wishes to take? Is it innovative? Tell us a little bit about whether the investigator is well trained to be able to carry out this work, and is she working in an institution, an environment that will enable the work to go further. Every reviewer has to comment on each of these 5 criteria, and then to be able to give a score, excuse me. So scoring of the NIH grant application by each study section, the grants are scored 1 through 9, exceptional to poor. And then all the scores for each of the 20 or so members of the study section are averaged and multiplied by 10. So the final outcome is a score that has 81 gradations from 10 to 90. And then those scores are then aligned in order and funded. And some of those grants are then funded. So the peer review process has some complexities, and it faces complexities, but it does so really well. So NIH peer review has to manage intrinsic conflicts of interest. It's the scientists who hold NIH grants that are evaluating the applications from other scientists who wish to hold a grant. So there's already an intrinsic conflict there because there's only a single pot of money. Those conflicts are managed very, very well in the process. I've been through decades of sitting in these peer review panels and watching how they operate. It operates on a massive scale, 80,000 grant applications per year across a broad scope, and incorporates lots of different metrics that we just looked at for merit and success. Not only that, the peer review system has to look at different types and styles of research, any and all of which can merit support. The different types of research that we've talked about, basic, translational, clinical, and population research. There's some grant mechanisms that take a linear step forward and deepen our understanding of the current paradigms for the ways that we think about how biological processes work. There are others that are transformative, that say, this is a crazy way that we've been thinking about this. Here's a whole new way to think about it. Very bold and transformative, obviously much lower chances of success, but if success is seen, the impact will be enormous. And technology, increasingly, scientific research is driven forward by new methods and new instruments, machines that are able to see things better and faster and higher resolution and so forth. And grant applications come in that make proposals of that sort. But from that complexity, remarkable things come. So it's not a perfect system, but amazing things happen. So here's one that you're all aware of. You've all heard of human genome editing that's been enabled by this amazing technique called CRISPR-Cas9, right? So I'm not going to try to explain how that works, just to tell you that remarkable tool that has completely revolutionized the way that biomedical research is carried out and is well on its way to revolutionizing the way that healthcare is dispensed and cure of disease will come into view, came from basic science research grant applications. One was simply to sequence the bacterial DNA, the DNA of bacteria that are able to grow in these incredibly disfavorable conditions in acidic mine drainages. And so the NIH said, okay, let's look at the sequences of those bacteria. And then later, a discovery was made that showed that these funny sequences in the bacteria that grew in those drainages represented a bacterial immune system. It's an immune system that's not related to our own. And so you might say, oh, well, we shouldn't study that. That's a waste of time. We got to study our own immune systems. But in fact, NIH went ahead and funded those. And from those basic discoveries came this new CRISPR technology that has really, really revolutionized the world. So what's the alternative? And this peer review system, this complicated system that I've described to you, the best system? Is there a different way to do it? Coming from these best NIH grant mechanisms, the bottom-up investigator-initiated researches. Well, what could you think about? You could think about alternatives, some of which are in place in other countries in the world. Central planning of research topics and projects, so top-down instead of bottom-up. Really hierarchical control of the processes by the oldest guys in the room, and literally guys in the room, depending on the title, how much power they hold, how much seniority they have. Or you could say, no, let's just take the money and spread it evenly out across the research community and give everybody a little bit. So there are some alternatives. And you can see from the way I'm talking about it that they don't work. By any measure, NIH peer review is not the best good way to allocate resources. It's really the only good way. So NIH peer review identifies the best science. That's by any measure, as I said in the last slide. And you could do that by the scientific publications or the advances that have been made, the big prizes that have been given for big discoveries, or the actual impact on health and the effectiveness and efficiency of the research as it is carried out. What's clear is that the ongoing commitment to excellence offers continued promise for really remarkable progress in scientific research and development that really turns into real impact. Nevertheless, paying for all of that great science is a problem. That peer review process that we described that filters through the quality of the research, at the end of the day, only 1 in 7 of the grant applications that are judged to be meritorious actually it receives funding. Because while $41 billion is a lot of money, it's not enough to be able to support all of the good research, all of the great research that is proposed. And it means that there are opportunities and advances that are lost. Nevertheless, let's face it, $41 billion is still lots of money. And so it's very fair for you, in fact, I think it's incumbent on you to ask, is there a return on investment? Are we doing this in the right way, right? And your constituents could be asking you the same question, right? Fair enough, important question to ask. And here's where I suggest that you impose something that I call the Starbucks test for your constituents. Because $41 billion are numbers that you're used to thinking about in Congress, but the average person doesn't really have any kind of conception of what that means. And so when you ask, is the money being spent efficiently, it's really hard to try to answer that question. So that's when I say, give them the Starbucks test. So here's the Starbucks test. I'm going to give it to you. How effective and efficient is NIH research? So fact, over the past 40 years, cardiovascular disease mortality has declined by 60%. This means 1 million fewer deaths per year. not bad. So here's the Starbucks test. What was the NIH investment per American resident per year in $5 Starbucks beverages that saved those million lives per year? Okay, so write in your blue books, the answer is one $5 Starbucks drink per year per resident over the past 40 years has now saved 1 million lives per year, 1 million people that would have died of cardiovascular disease. So the question for your constituent is, would you give up 1 Starbucks per year to save 1 million lives? And just think of what could be done if we gave up 2. So there we are. This is the NIH, the crown jewel of federal spending, as some people call it. I think it is a representation of federal government at its best in being able to highlight quality, efficiency, and impact. And it carries out those or achieves those goals because of the volunteer efforts of scientists who come to Washington, volunteer their time to identify the best research proposal, and because of you, of Congress, that provides the funding to allow that work to go forward. So as Lynn described or mentioned at the outset of this session, tell your members to join the Congressional Biomedical Research Caucus. There's the URL, and she has it in the chat as well. And it really means that you can be involved in supporting and increasing funding for NIH research to institutions and scientists and trainees in your district and states. And I'll stop there and be happy to discuss and respond to any questions you would have.
Thank you, Keith. That was a great talk, and I always love the Starbucks analogy. We do have some questions that have come in, and I do encourage folks to ask their question in the question and answer box located at the bottom of your screen. The first question is, I am a parent of a child who has both rare and undiagnosed disease. My question is twofold. Are there ever considerations to create new institutes? And to that, has there ever been consideration for an institute for rare and undiagnosed diseases?
It's a great question, a very important one, obviously, for the questioner, but really important for all of us. And I'm going to give you a response that may sound contradictory at first. And that is that it's my view, and it's a view shared by some others as well that are involved in science policy, that in fact there are already too many NIH institutes, and that we wouldn't be looking for new ones. But you say, why in the heck is that? Why can't we put focus on rare diseases that are important? There's lots of rare diseases, several thousand actually. The reason I have that view is that as we've learned more about the disease, we've understood that in fact diseases that have one name actually have multiple causes, and those different causation pathways, the different pathways through that plate of spaghetti that I described in that one slide, are under investigation in a number of different NIH institutes now, because they've been discovered to actually act on one kind of disease. But what we're finding is that they're also instrumental in causing others. So one disease actually has many causes. And the converse is also true, that a cause for a disease can actually, an alteration or mutation can actually be responsible for causing multiple diseases that don't look like each other. My favorite example is a group of diseases called Ciliopathies. These are diseases of defects in cilia, and the cilia is a simple little hair that actually extends off of each of your cells, right? That is involved in a number of processes. And it turns out that defects in cilia are responsible for a series of diseases that don't look like each other. Hydrocephaly, polydactyly, polycystic kidney disease, certain forms of obesity, asthma. Again, scientists out in different institutes are studying these diseases. And in fact, what they're studying are processes that are all driven by the same defect, right? And so by separating the scientists out into these separate institutes and the support for them in separate institute budgets, we are sacrificing the kinds of interactions that could bring knowledge to a range of diseases that we didn't suspect before were involved, and that would certainly include rare diseases. The second way to think about this is to go back to that precision medicine approach that I described. And you remember that the idea of precision medicine is to bring together all of these different kinds of information and to integrate them and analyze them in ways that will allow us to be able to distinguish and determine how one person's disease is different from the next person's. And I mean this, even identical twins are going to have different, because they've had different experiences and exposures and life histories and behaviors and so forth. So really what this says is that as we understand precision medicine better, every disease will become a rare disease. We really need to accommodate that in the ways that we think about our research, the ways that we develop drugs and so forth. So I would say, rather than standing up a new institute for each rare disease, we better integrate the information across the institutes. And one might even think, in the science policy sense, one might even think about reducing the number of NIH institutes so that they're more interactive, the scientists within them are already more interactive.
We have a couple more questions that have come in. What are your thoughts on creating a Harper, a Health Innovation Accelerator at NIH?
So this is an interesting idea that has been put forward and really capitalizes on an approach, an organizational approach for research that was generated within the Department of Defense by an agency called DARPA. So the Defense Accelerator, and is in which projects are very strongly managed, unlike the way that NIH grants are administered, where the investigator comes in, tells the NIH, here's what I want to be able to do, please give me some money to do that. And NIH says, fantastic application, here's your money, you'll have money for 4 years, 5 years, and just kind of check in with us every year to let us know how things are going, and we'll give you your next aliquot of money. Instead, the DARPA and Harper approach is for the agency to be able to determine a specific project that they really want to be able to move forward, and find scientists who, using their imagination and creativity, propose specific innovative ways to try to move toward that end goal. So much more application in mind. And I think that there is room within the NIH for standing up an agency within the agency, so to speak, to allow that kind of research approach, managed research approach, to be taken. But it will be important to be able to do this in a measured way, to realize, as we said, that there's still way more that we don't understand than what we do understand. And so while there's room for targeted research that we know about, and one could think about what happened with COVID here, that it's important for the bulk of the research, that pyramid that we described, to still be really driven by basic discovery that is curiosity driven, and that the buildup of those facts together will then also move us toward applications that we couldn't otherwise already have imagined. So the DARPA-Harper approach really moves toward application through pathways and mechanisms that we already do imagine. That's how you develop a targeted research approach. But to still leave lots of room, actually the bulk of the room, for approaches and concepts that we haven't not yet imagined.
let me ask this next question. It was very interesting to learn about trials in less complex species. Could you share any information on the use of modeling and simulation at the NIH to better predict long-term side effects for treatments?
Great question. So I think that in a way, the precision medicine approach embodies the approaches that the questioner is asking about. Because in one sense, the precision medicine, the culmination of precision medicine, is a big computer science problem that says, how do we compare facts that come from a population study to look at the incidence of disease in a given zip code, in a zip code that was affected by wildfires, and they had 3 or 7 weeks of exposure to high particulates in the air, and looking at that against the molecular mechanisms that we're studying, against the genome sequence of individuals. This is a big machine learning and artificial intelligence puzzle that computer scientists really need to work out. And it's really through those kinds of grounding approaches that are being developed by those that are really pushing the envelope on precision medicine that we're going to be able to bring together the kinds of information that the questioner is asking about. And that's where the big, really high-end modeling approaches, I think, are going to really bear fruit.
We have one more. How does NIH get its scientists among the institutes and centers to better interact and share information?
Great. It's a great question. I think that the federal budgeting process that all of you are involved in is a problem here, and we need to work on it. So the questioner is exactly right that across the different institutes of the NIH and the scientists working under the support of those institutes around the country in your districts and states are working in truth in silos. The silos are defined by the budgets that go to the institutes, and every year the institute, each of the NIH institute directors has to go to the Hill, to your hearing rooms, and defend the budget of the National Institute on Drug Abuse. And the way that they do that, the way that they're essentially forced to do that, is to say, well, the work that's going on at NIDA, National Institute of Drug Abuse, is completely unique. There's no other NIH institute doing it and no other federal agency doing it. And so if you, congressperson, shut off my budget, right, all of that progress will stop, right? And so you have to keep giving me money. So it's a natural argument. And the way that the institute director can defend that is to not interact with the other institutes, to not interact with other federal agencies that are involved in work that could contribute to the progress. So then the director can say, no one else is doing this. It's completely unique. You have to give me my money, right? And so the scientists interact less and less. NIH has institutes within it where they have computer systems that don't talk with each other. They're not interoperable, and everybody stays in their lane, right? This is not quite as bad as I'm making it out to be, but it is essentially like that. And certainly cooperation across agencies is very poor. The Health and Human Services houses 3 critical health agencies, the NIH, we've been talking about, that you're aware of all of them, the CDC and the FDA, right? And again, there's not as much, there's relatively little, to be honest, interaction across those agency boundaries. So I think that you in Congress could do a lot in your budgeting process. The authorizing committees could put in language that would encourage cross-agency cooperation in ways that could really be effective. In the Science and Technology Action Plan that I flashed a slide for, we made a specific proposal that the Office of Science and Technology Policy, the OSTP, where Eric Lander is the designee as the director and will be a science advisor who President Biden has elevated to cabinet level, that the OSTP would house a set of coordinators whose job it would be to look at the problems at hand in health and healthcare and public health, for example, in food and water security, in energy utilization, production and storage, and in the environment and climate change. Look at those big problems and say, what agencies should be working together to be able to make some movement on these huge problems, right? And that their job, they would have the authority to ask agencies to come forward with joint proposals in which scientists would be working together. So we hope that as the science technology plan moves forward into legislation, that could be one way to be able to lead to better interaction across what right now remain as pretty sturdy silo boundaries.
Could you give an example of how NIH researcher may work with other agencies like the Department of Energy Office of Science Laboratories?
So give an example of how
an NIH researcher may work with other agencies like the Department of Energy.
Yeah, it's not simple. There are relatively few joint programs like that. The questioner asked, picked out two agencies that could really benefit from working more effectively together. We're fortunate. I happen to be on the advisory board for the Lawrence Berkeley Laboratory, which is a DOE national laboratory, which is the one national laboratory in the 17 DOE national lab network that actually has a biosciences directorate, and so we're able to work well at being able to stand up joint projects that across those boundaries, but it's not easy. That because of the concerns about the way the budgeting is set up, it's not an easy discussion to have to be able to stand up joint projects. So what we've done at UCSF is to build specific funding programs in which we put in a certain amount of money. Actually, right now we have a tri-institutional program where UC Davis, UCSF, and the Lawrence Berkeley National Lab has put in funding to stand up and support research projects on the microbiome. So we've basically done it internally to build our own programs that will allow these scientists to work together, because we know that if they collaborate, great things will happen that wouldn't happen individually.
This is more of a follow-up to the previous questions, but can OSTP help with the integration across federal science agencies?
Yeah, I think great question, and I think it's the only way because of this blockade that's imposed by the budgeting process. And so what we have proposed in the Science and Technology Action Plan is for the OSTP to have $1 billion that these coordinators that are charged with bringing together scientists across agency boundaries, so that when the OSTP director goes to the Department of Energy and the NIH and says, come back to us with a plan, a project that will begin to use the high-end computation skills and hardware sitting in the DOE national labs to build a knowledge network that will bring together all of those health data, health and biological research data that we talked about in an effective way for actual analysis using machine learning and artificial intelligence. Come back to us with a plan for doing that. And if we like it, if we like that plan, we have money that we will give you to supplement your own budgets, right, to enable that interaction to happen. So I think we need to have a carrot as well as a stick. And the Science Technology Action Plan acknowledges that and builds funding into that process.
So we had a follow-up question from our attendee who asked about rare and undiagnosed disease, and she goes on to say, wouldn't an institute looking at rare and undiagnosed disease where there is really an intersectionality of disease be a good idea where doctors and scientists partner across specialties, where they are not working in silos but in partnership?
Yeah, it might be that you would have an institute of that sort. It's not clear how one would actually determine the resources that would go into that institute as opposed to others. And what the questioner is proposing is bringing together, having a mechanism that would bring together scientists who are working on disparate biological processes and disease processes to work together in a team, and to have the support for that process under the roof of one new institute that would be stood up. I can imagine doing that. It's hard to think of how you would actually set the budgeting for that. What the hope would be, and I suspect that this is the hope of the questioner as well, is that more and more teams of that sort would form and come together under this new umbrella to support exciting research in which the investigators are very certain that they are now working on a project that they could no way work on by themselves, but working in these teams, cross-disciplinary teams, they've got a real shot and making real progress. And I think that could happen. Whether it should be housed in a new institute of that sort, I think is the main question, as opposed to thinking of other mechanisms that will help these individuals come together. So I'll mention one, which is simply for the NIH to stand up a set of new funding mechanisms, new grant mechanisms that say, come to us as teams, of 3, 4, 5, 6 investigators that have different expertise, different approaches, different points of view about biological and disease problems, that in which you sat down with each other and realized that if you work together, you can really define a project that you wouldn't have been able to define by yourself, much less actually make progress on it. And I think that could be done. So simply standing up a new way of supporting scientists might be enough to be able to motivate, because the scientists are out for money. They want to be able to study projects of their interest, but if the NIH shows no interest in being able to support that, then that idea that they may have in the shower will just go down the drain. But instead, if you have this enabling tool saying, yes, we have a funding mechanism for you, that might work. But I think that'll work better than standing up a separate institute. But the questioner is right on in being able to say, let's struggle together to think about ways to be able to do this, because it's really what we need.
Okay, and I think this next question might be our last, given our time, but we'll see. And it's a toughie, and it's one that we could probably have a caucus conversation on by itself. But the questioner asks, it is great we can make discoveries, but how do we protect our investment from foreign espionage?
Yeah, this is, I'm sure the questioner is aware that this is a timely question, that we are, I think that we are happy to have arrived at created an environment in which scientific research is a global endeavor, and that everyone who's involved in it realizes that we're going to move forward better and faster if we maintain this global network of interaction of scientists. At the same time, different countries have different standards by which they're operating for being able to capture and realize the fruits of new intellectual property as it is discovered and developed. And so there's a struggle going on to find, to put it benevolently, a balance between the kind of security for discoveries that can turn into marketable products and products that help people, and simply having an open marketplace of ideas and information. So the more open we can make our science, the better the science will be. But in fact, being able then to move toward application involves intellectual property protection in which the standards differ across different countries. And people are all aware of this, and I think that all we can do is to in fact, one of the reasons that it is important for the United States to maintain primacy in scientific research and development is that the leader gets to have the biggest voice in enunciating standards and being able to enforce values. And so if the US is able to maintain its intellectual lead, it will also be able to be a leader in enunciating the kind of qualitative boundaries and values and standards by which that information is actually used. So we can't go to another country and say, here are the IP pathways that you should follow. But we can say, here's what we're doing and why we think this is the right approach. This is exactly the approach at the scientific level that we've taken with stem cell research, with human genome editing research. The national academies pulled together other national academies around the world to be able to develop these things jointly. And the pathways that were developed were actually adopted or adapted, at least adapted, by not only those countries that are involved in setting those standards, but by others that were not. So you know, I'm cautious, but cautiously optimistic that we can do the same thing with intellectual property. It's right now an ongoing struggle and something we're just going to continue to need to work on. Lynn is right that this could be a topic of very much of its own for a caucus gathering.
Okay, with that, I'd like to wrap up. I want to thank Congressman Curtis for joining us, and a special thanks to Dr. Yamamoto for his presentation. So thank you, and I hope you all have a great day.
Great, thank you. Take care.